morning, all. I am Dr. A. Dayanand, Assistant Professor of Chemistry, Government City College, Hyderabad. Respected Principal, Sir, and Patron of this session, Dr. P. Balavaskar Garu. Today's distinguished speaker, Dr. Mahender B. Dewal Garu, Head R&D, Founding Member, Expansion Technologies, Inc., Boston, USA. Dr. Viplava Shukla Garu, HOD Chemistry, and distinguished professors of Usmania University, former professors and heads of the Department of Chemistry, Usmania University, and all our colleagues, staff members of Government City College, teaching faculty from various colleges, and student participants. A warm welcome to you all to the international webinar on living with COVID-19, role of therapeutics and vaccines to overcome the pandemic. This session is organized, and it is our pleasure that principal sir had accepted to organize this session. And this session is organized mainly that we had seen in the first wave and second wave, many lives were lost, and many livelihoods they had lost, even their livelihoods also. Why? Because a new normal should be followed, but the citizens, either students or the science community, are unaware of the new normal. Therefore, this session is organized to sensitize the new normal aspects as well, and different areas where we had to focus. Thank you, sir, for accepting our invitation, Mahender sir. And our city college is the oldest college. It is having 100 years of legacy. It is the oldest and the topmost that is prioritized. Students select this college as the first priority while admissions also. We are in the forefront, as you are in the forefront as a warrior against COVID. We are in the forefront of giving the education to the students. Now I invite patron of the webinar, Dr. B. Balavaskar Garu, to address the gathering.
Thank you, Dayanand. Good morning to all the members and the participants. First of all, I must thank today's resource person, Mahender Dewal Garu, for accepting our invitation and coming on this webinar to share his views and knowledge with us. And I also welcome all the other chemistry senior and present faculty members who are all the stalwarts are there in the meeting now, that I understood from the gathering. So for this, I invite them, I welcome them for this webinar. I also congratulate our chemistry department head Viplava Shukla Garu and other senior faculty members Yadaya Garu and Dayanand Garu, Bussi Anil Garu from the chemistry department, Vasanta Garu, for organizing a wonderful webinar of this kind. Definitely, this is a topic our department has chosen, a very apt topic for the present situation. We all been observing and noting that the first and second wave created a disaster in the entire globe. But the third wave, of course, it is there, but it is not giving that much of damage to the society, to the mankind. The reason many of the people the experts are saying that because of the vaccination, definitely this has been controlled. So definitely it's a very apt topic that our department has chosen. I hope our resource person is going to share his views and throw a light on it. Definitely it will be going to useful for our students also. I request our department to record the entire program so that this will be shared on our college website to the students also, whoever not joined in the meeting. We can also share the link for those students. That will be very useful for them. I once again thank the resource person especially Mahender Garu, for accepting and joining the webinar from Boston, USA, and also the senior chemistry faculty who are retired and who are in America and who are with all different walks of life. But they also spared their valuable time to be with us today for this webinar. I thank all the participants who are there in this meeting. And I once again congratulate all our chemistry department faculty members. Definitely this webinar is going to be useful for our students and to the community in particular. Definitely, I hope this will go in a good way. Best of luck to the webinar. Thank you all.
Thank you, sir. Thank you. It is live recorded, sir. Already the live recording is on. And now I request Srimati A. Vasanta Garu, Assistant Professor of Chemistry, Government City College, to introduce today's distinguished speaker, Dr. Mahender B. Dewal Garu.
Thank you, sir. Thank you, sir. And respected principal, sir, and today's honorable guest, Dr. Mahender B. Dewal, and distinguished guests present in this webinar, and my dear colleagues and my dear students, a very good morning to all. And it is my honor and privilege to introduce Mahender B. Dewal Garu. Dr. Mahender B. Dewal is a biomedical scientist and biotech entrepreneur with 20 years of interdisciplinary experience in number of research areas, including organic chemistry, medicinal chemistry, biochemistry, molecular and cellular biology, chemical biology, antibody engineering, genomics, transcriptomics, and expansion microscopy. And about his studies, Dr. Mahender B. Dewal received his bachelor's with chemistry and biology as a majors from Giriraj Government Degree College, Nizamabad, and a master's degree in specialization in organic chemistry from Osmania University campus, Hyderabad. And he has received his PhD in organic chemistry from Department of Chemistry and Biochemistry, University of South Carolina, Columbia, South Carolina, USA. And he has completed his postdoctoral studies from Department of Pharmaceutical Sciences, Warne State University, Detroit, MI, USA. He has worked as a research scientist at Department of Chemistry, Massachusetts Institute of Technology, Cambridge, MA, USA. Presently, Dr. Mahender B. Dewal is the founding member and head of research and development at Expansion Technologies in Cambridge, Massachusetts, USA. Dr. Dewal also is an affiliated to world's renowned Massachusetts Institute of Technology, Cambridge, USA. His research lab has been working on developing early detection tests for various cancers, including breast, prostate, lung, and brain, etc. However, as the COVID-19 pandemic hit the world in December 2019, Dr. Dewal's team started working on developing antibody therapeutics for COVID-19 infections. Additionally, from the past 1 year, Dr. Dewal has been actively engaged in giving international webinars and participating in many debates in Indian news channels to raise COVID-19 awareness, such as viral infection mechanisms, precautionary measures, and diagnostic tests, vaccine, and therapeutic developments. Dr. Dewal spoke about the challenges of a post-COVID-19 pandemic, a 24-hour World Book of Records event organized by Lead India Foundation, a brainchild of our beloved 11th President of India, Dr. APJ Abdul Kalam. Dr. Dewal's service on COVID-19 fight had recognized him as COVID-19 warrior by together Karnataka State Ministry of Youth Empowerment and Sports and SAARC Nations International Human Rights Association. His research was published in high quality and well-reputed journals, including Journal of the American Chemical Society, ACS Infectious Diseases, Chemistry and Biology, ACS Chemical Biology, Proceedings of the National Academy of Sciences of the United States of America, etc. His research was featured in Research Highlights of Nature Chemistry. Dr. Dewal also presently holds 4 US and international patents. Dr. Dewal has been president of Bonn State University Postdoctoral Association for the year of 2012, also served as an advisory board member and organizing committee member as well. Dr. Dewal also has been vice president of it. MIT Ventureships Club and ASA recognized independent student group aimed at providing MIT students with working experience in startup companies, and also served as an advisory board member. And Dewal has been a member of American Chemical Society and peer reviewer of many international journals. So with this small introduction, I am inviting to deliver lecture Dr. B. Dewal Garu, Mahender B. Dewal Garu. Please, I am handing over to the sir.
Thank you so much, Vasanta Garu, for a very kind introduction. Good morning, everyone. Good morning, good afternoon, good evening, everyone. It's a pleasure for me to be here today. Thank you so much for organizing this event. All the City College Department of Chemistry, faculty members and students, and inviting for me. It's a very glad to see my professors and not only the faculty members. I think some of my alumni members also is there. It's a great pleasure for me to see all of you here at this stage. Without a further delay, let me share my presentation and then we'll get going. Can everybody see my screen?
Yeah, we are able to
see the screen, right? And you can hear me well?
Yeah, it's good.
Very clarity, no problem.
Thank you so much. So today I'm going to talk about living with COVID-19, the role of vaccines and therapeutics to overcome the pandemic. So first, so it's been almost 2 years we have been living with this COVID-19 pandemic, right? It started around November, December in 2019 in this place in China as a mysterious virus infection in a place called Wuhan. By January 2020, the first death was reported. Then by March 2020, it spread pretty much many countries all over the world. And you can see that the world map behind here. So the World Health Organization declared it as a pandemic. But by May 2020, we were able to get one of the therapeutics, Remdesivir, approved by Food and Drug Administration at United States for emergency use for the critical patients. By September 2020, within about 10 months of the pandemic, or maybe 7 months of the pandemic declared by World Health Organization, the death rate reached to almost 1 million all over the world. And the good thing is, by December 2020, within a 1 year of time, we were able to get successfully able to get 2 vaccines, novel kind of vaccines, were able to authorized as an emergency use to fight with these coronavirus infections. One of them is Pfizer-BioNTech vaccine, and the second one is Moderna vaccine. However, Everybody thought that when we had the vaccines, it's going to stop, but it's not stopped. It's continued in the form of mutations. Here you can see these, it's a Greek alphabet. I put it here because we are calling all these mutations with the Greek alphabets, right? Alpha, beta, gamma, and delta. There are so many in between now. Omicron, one of the very infectious variant, emerged, and we don't know whether many more are going to come in. It's still like around 9 alphabets: pi, rho, sigma, tau, upsilon, till omega. Some more alphabets left. Once the Greek alphabet is done, we don't know. Go to the next alphabet. I'm not sure. Hopefully, we're not going to go over that. But you can see that here, on this plot, number of waves all over the world. You can divide these as waves, right? Number of cases reported, approximately 4th wave, around like more than 3.5-4 million cases in a 1 week average, a daily average of 1 week throughout the world. If you look into the US, this is almost like the fifth wave is going on right now. Approximately around 800,000 cases, daily average of 7 days. And in India, now you can see that the 3rd wave is going on with a daily average of 300,000 cases of weekly 7-day average. So it all started with this small piece of virus. This is a transmission electron micrograph of the SARS coronavirus 2 particles. These are orange particles on the surface of a cell. And you can see the much more close-up structure here. And the one more important thing I would like to say here is the virus can be visible only some of the very high microscopes like transmission electron microscope, super resolution microscopes, but not with any other X-ray scannings or CT scans or computer tomographies or any other type of scannings. The virus is not visible. This is the much more closer look, the real coronavirus extracted from, collected from the humans that are infected. So this is a computer animation. You can see that here, this virus, coronavirus, there is a positive RNA genome. It has a positive sense RNA genome, which is securely, you can see this orange color positive sense RNA genome, securely secured by 4 structural proteins. First one, these green ones are the spike proteins. And there is a nucleocapsid protein, which is protecting the genome of the virus. And the yellow ones are the membrane proteins. And the red ones are the envelope proteins. So this is a 2D map of the virus genome, the virus RNA, the organization. Around 29 to 30,000 nucleotides together form this RNA. And it is divided into 2 parts. First part has 2 open reading frames, which encodes approximately 16 non-structural proteins, where they are called NSPs, which are much more crucial in the function of the virus, in the virus production, virus replication. And the rest of the part has 4 structural proteins, as I mentioned in my previous slide, including 7 accessory proteins. However, among all these proteins, the spike protein, which plays a very important role in the virus infection. If you look at here on the left side, this is the virus membrane. The trimer of the spike protein came out here, and this is the ACE2 receptor on the human cell. The virus spike protein binds to the ACE2 receptor of the human cell. There are 3 ACE2 receptors of the trimer of the spike protein is binding. It bounds very strongly, kind of clicks into that ACE2 receptor. Once it is clicked, these orange and yellow color human proteins, proteases called proteases, they cleave the 2 subunits of the virus spike protein into 1 subunit, which is the S1 subunit. After that, this S1 subunit go through some structural changes and then attach to the human cell membrane, and then it started fusing into the human cell membrane. And then once it fuses, then it will releases the RNA, the virus genome into the human cell. Once it releases the human cell, this process is called an endocytosis process. Once the genome is released into the human cell, it started the first open reading frames, first half, the non-structural proteins are formed immediately, including the RDRP. RNA-dependent RNA polymerase, one of the crucial protein for the RNA replication. The second one is the proteases will be released, and then they generate all the subgenomic RNAs, which encodes the structural proteins, accessory proteins, rest of the non-structural proteins. And then once the structural proteins are created, they go into the Golgi and endoplasmic reticulum of the cell, where they mature more. They get matured, post-translationally modified, and then the virus started assembling at the membrane of the either Golgi or the endoplasmic reticulum. And once the complete viral particle, functional virus particle is formed at the membrane, then it will be transferred towards the cell membrane, and it will be released into the outside of the cell so that it can infect to more and more cells. This process is called exocytosis process. Once the virus infects the human body, the first protection system that will be reacted is human immune system. There are two types of immune systems. The first one is called innate immunity. The second one is called adaptive immunity. Every human being by birth contains this innate immunity. They're born with the innate immunity. This is more of a frontline defense, which reacts non-specifically. As soon as something happens into the body, it will be activated, and then it within like in hours, less than 12 hours, it will be activated and then releases the protection mechanisms. The adaptive immunity, it is kind of a slow response. Its response a bit slow. It's kind of a backup and a defensive force. It specifically responds, though it is responded within a days, like slowly, but the response stays from months to years, long periods of time. Both these two, innate immunity as well as the adaptive immunity, work together. They talk together to protect the human body from the toxins or the virus infections or the bacterial infections, any type of diseases as well. So the innate immunity has so many different types of cells, but I'm going to explain here only a couple of them. Excuse me. As soon as it sees the virus infection, it releases various different chemicals called cytokines and chemokines. These cytokines and chemokines try to find the infection or the toxin. They go there and they try to reduce their impact. As soon as the cytokines and chemokines try to reduce the impact, the macrophages, another type of cells, they go there and they engulf the virus particles. While they're doing this, their part, the nature killer cells, they try to activate the suicidal mechanisms in the cells that are infected by the virus. So the cells will be dead, and then the virus will be cleared out. While these two are doing their job, another cell type called antigen presenting cells, which are also called dendritic cells, takes the antigenic molecules, the exclusive molecules that are present on the virus or bacteria or whatever it is. For example, for the COVID-19, the spike protein is the antigen molecule. They take these molecules and runs towards the adaptive immune system. In the adaptive immune system, the antigen presenting cells gives the information about the antigen molecules to the T cells. T cells differentiated into two types of cells, T helper cells and cytotoxic lymphocytes. T helper cells generates another cell, which is B cells and plasma cells as well. These B cells produces lots of antibodies against that particular antigen. For example, COVID-19, a spike protein. The antigens against the spike protein or other nucleocapsid protein, membrane, any type of protein against to any type of protein, they produce number of antibodies. And these antibodies are also called neutralizing antibodies. What they do is they go and they surround the virus, and then they neutralizes it so that it will not infects to the new cells. So that's how they clear the body. The immune system clears the infections or toxins or the bacteria, diseases out of our body. While immune system is working very well when the coronavirus infects, but the coronavirus is kind of somehow it's hijacks the immune system, and it tricks the immune system, it escapes from the immune response, and it generates. Can somebody mute, please? There is a lot of sound coming. Thank you. So it releases lots and lots of cytokines and chemokines, which is called cytokine storm. These cytokines, instead of killing the virus, they attack the human. So for coronavirus, which infects the lungs, they're attacking the lungs, and the lungs are lung cells are damaged. When the lung cells damaged, lung tissue will be damaged, and then it will start the disease progress into severe disease. So when the virus infection is not being controlled by the immune system, the human immune system, then at that point we need some kind of additional protection, which is the treatment option. And also vaccines. So the vaccines, they're not going to cure the infection, but they work as a preventive mechanism, protection for the infection. They as soon as they creates additional force in the human body in addition to the immune system, so that as soon as the virus gets inside the body, it will be cleared out. So first, I'm going to start discussing about the vaccines, and then we'll go into the therapeutics. So let's look into the how does this vaccines work. Please mute. I can't really. This is I put the headphones so that noise, somebody's phone or somebody's. Please mute everybody except me, because this is giving me a lot of noise. Thanks. So yeah, the vaccines strengthens the adaptive immune system. As I mentioned in my previous slides, the adaptive immune system responds slowly, but it stays long period of time. It strengthens the adaptive immune system. What does it do is, the vaccine is nothing but, it could be a virus or a part of the virus, a part like a spike protein or something else, or its DNA or virus genome or virus protein or anything, or virus-like things can be act as a vaccine. When it is injected into the human body, the antigen molecules on this vaccine will be recognized by the antigen-presenting cells or the dendritic cells, and they take it to the T cells in the adaptive immune system, which will generate the cytotoxic T cells, as well as the B cells. B cells produces a number of antibodies, neutralizing antibodies. In addition to the cytotoxic T cells and B cells, the vaccine generates the memory B cells, memory T cells, and effector T cells. What these memory B cells and memory T cells does is, they remembers the mechanism of the neutralization mechanism of the virus and virus antigen particles of that particular virus, whether it is a coronavirus or it's a flu virus, it's an Ebola virus. Every virus has its own memory cells. A vaccine to every virus has their own memory cells will be generated. This is how the vaccines work. Here I've listed various different approaches to develop the vaccines. So there are three different approaches. Classical approaches, where the virus itself will be utilized. And second approach is conventional approach, where a part of the virus, like I said, protein or some other parts of the virus or virus-like particles can be utilized to develop the vaccine. And the third approach is next generation platforms, where virus genome, like a nucleic acid or virus vectors, different other viruses can be modified to use as a vaccines. These are the three approaches. And to develop a vaccine, the antigenic molecules are very crucial, very important. And particularly for this coronavirus, the structural proteins, spike, envelope, matrix, nucleocapsid proteins could be very good antigen molecules to develop the vaccine. Approximately around 234 candidates are under development, and among the 234, 140 of them are already in the clinical trials. One of the vaccines is completely approved, and approximately 10 to 12 vaccines are currently used under the emergency use authorization all over the world. In the first approach, there are two methods: the virus-based vaccines, classical methods. Live attenuated virus, that means a weakened virus, can be utilized as a potential vaccine. So that means the virus that, let's say, the smallpox virus, measles, mumps, rubella virus, chickenpox virus, influenza virus are developed using this method. What they do is, this virus will be cultured in the cells, human cells or the human animals, for several generations until it will be completely weakened. It will completely lose its infectiousness, its activity. And then at that point, it will be utilized as a vaccine. There are only two potential companies. Serum Institute of India, together with Quadrogenics, which is called Covivax, is developing a vaccine using this method, and also Mesa Vaccines also developing one more vaccine. Only two among all those 234 candidates are these methods are developing vaccines using this particular method. They're in the phase 1, 2, 3 clinical trials right now. The second method is using the inactivated virus. That means killed virus. The virus will be inactivated using some chemicals like formaldehyde or propanaldehyde or heat, some kind of, or culturing them in the egg, something like that. The virus will be inactivated or killed, and it will be utilized as a potential vaccine. Polio vaccine, hepatitis A vaccine, rabies vaccine. These are the best examples that have been developed using this particular method. For the coronavirus 19, our vaccine, the first indigenous vaccine developed by Bharat Biotech, is the inactivated virus. They took the coronavirus, they inactivated using beta propia on the head, a chemical, and then they made it with a couple of other adjuvants like imidazopyridine and aluminum hydroxide gel. Together, they made this vaccine. It showed a very good efficiency. Right now, it's being it's being used all over the world under emergency use authorization. 2 other vaccines developed from China, Sinovac and Sinopharm, also in the same form against these COVID-19 coronavirus infections. Other approach is a conventional approach using the virus-like particles. So the viruses are kind of modified like a virus-like particles. These virus-like particles are not real viruses. They look like a virus. So HPV vaccine, hepatitis B vaccines are developed using this particular method. Spy Biotech, Medicago, these are with the Serum Institute of India. These are the couple of examples that I gave here are using a method to develop a vaccine for coronaviruses. And protein-based vaccines, particularly for the coronavirus, the spike protein can be utilized to develop the vaccine or a part of the spike protein. A company called Novavax is developing using the spike protein of this coronavirus. And right now, a couple of countries authorized for the emergency use. It's still on the phase 3 clinical trials for the few other countries. And Serum Institute of India is collaborating with Novavax to develop this vaccine. As soon as it is cleared by DCGI, it will be available in India as well, which is with the name as a Covovax. And in the phase 3 clinical trials, this one showed about 89% efficiency among the 15,000 participants. Now, the third approach is using the next generation platforms. The first method here is viral vectors, using the viral vectors as a vaccine. In here also, in this approach also, there are 2 methods. Using the replicating viral vectors, for example, in the first method I have mentioned about live attenuated virus. That virus can be taken, for example, a polio virus can be taken and engineered their genome with the coronavirus spike protein genome and utilize it as a vaccine. So for the Ebola vaccine, they used recombinant vesicular stomatitis virus. They took the vesicular stomatitis virus and engineered the viral genome with the Ebola virus genome and then utilized as a vaccine against the Ebola virus. And it is approved by FDA in December 2019. This is one of the methods to develop. Israel Institute of Biomedical Research using this method to develop a vaccine, which is under phase 1 and 2 clinical trials. It's kind of a little rare method to make the viruses vaccines. The second approach is non-replicating viral vectors. That means this virus is live. Both the methods, the viruses are live. Here, the virus can replicate. The vaccine that made using this approach, it can replicate. It can make more and more inside the human body. But this one, non-replicating viral vectors, though this virus is alive, it cannot replicate. It cannot produce more and more itself. It just whatever the virus you give as a vaccine into the body, that will produce the immune response. Also, these are also genome of these particular viruses will be engineered with the virus that we are developing the vaccine towards. For coronavirus, obviously the spike protein genome will be incorporated. Rest of this virus's genome will be deleted immediately so that it will not cause any harm to the body. So once you inject this vaccine inside the body, it will activate the adaptive immune response to produce the antibodies and memory cells. See, here are some examples that are developed using this approach. Non-replicating viral vectors, mostly Oxford AstraZeneca vaccine, which is one of the first generation COVID-19 vaccines. They have used chimpanzee adenovirus vector and engineered with the spike protein of the coronavirus. It is a 2-dose vaccine together with BARDA is American funding agency. They developed this vaccine together with these 3, AstraZeneca company, Oxford University, and BARDA, which showed a very good efficiency, and then it is authorized for emergency use in India, and it is being produced by Serum Institute of India with the brand name Covishield. And Consumer Biologics is one of the companies, the first company. This company, by March 2020, they came up with initial preclinical data. They used human adenovirus type 5 vector engineered with the spike protein. It's called Convidacia. This is a single dose vaccine, and it's been authorized for authorized in several countries for emergency use, which also showed a very good efficiency. The other vaccine using the same method developed by Russia, which is called Sputnik V, Russian direct investment fund together with the Gamaleya National Center. This is a 2-dose vaccine as well. But this vaccine is developed using 2 different human adenoviruses. First one is adenovirus type 26, and the second is adenovirus type 5. One of the virus is one of the vaccines that developed using this vector is given as the first dose, and the other one is the second dose, which showed a very good efficiency, and it's also authorized in India for emergency use and being distributed among the people as well. The other vaccine developed in this method is Johnson & Johnson's by Jennison, sister company of Johnson & Johnson. They also used adenovirus type 26, which is kind of similar to the Russian vaccine. It is also a single dose vaccine, which is authorized by FDA for emergency use authorization, which is being distributed in the US, showed a very good efficiency in the phase 3 clinical trials from 57 to 72%, depending upon the country, region, all over the world. The other method in the third approach, which is the next generation platforms, is using the viral genome as a potential vaccine. So it could be a DNA or it could be an RNA. For coronaviruses, the entire spike protein RNA or DNA can be utilized, or a part of which is a receptor binding domain. A part of the spike protein can be also utilized to develop the vaccine. DNA can be injected directly into the body to generate the immune response, but RNA has to be protected with something else so that it can produce the immune response. Because RNA is very stable, very unstable, it will be degraded very fast. The very first set of vaccines developed using the mRNA, the first one is Comirnaty by Pfizer and BioNTech together with Fusion Pharma. This is the first FDA completely approved vaccine for the 16 and above age group people, and the emergency use authorized for the above 5 year, between 5 to 15 age group children also. This is a 2-dose vaccine, showed very good efficiency. The immune response generated by this vaccine retains about 8 months. And it's currently the clinical trials are ongoing in children's less than 2 years, from 6 months to 2 years. The second vaccine, which is approved in December 2020, the Moderna vaccine, mRNA 1273. This is also authorized for emergency use in the United States for adults above 18 years. It's a 2-dose vaccine as well, so very good efficiency. Immune response lasts at least 6 months after the 2 doses. And this is also the clinical trials for the children are ongoing right now. The DNA-based vaccines, Inovio Pharmaceuticals is developing an INO 48800 is the vaccine candidate is still under the development. It's a 2-dose vaccine as well. The phase 3 clinical trials are ongoing. But in India, Zydus developed the DNA-based vaccine, which is called Zycov-D, which is authorized for the emergency use in the age group above 12 years. This is a 3-dose vaccine, which is given first dose, second dose after 4 weeks after the first dose, and then third dose 4 weeks after the second dose. So very good efficiency. So here I've listed several vaccine candidates which are in the front line right now, authorized for the emergency use various parts of the world, and approximately 6 vaccines: Bharat Biotech, Oxford AstraZeneca, Johnson & Johnson, Sputnik V, Novavax, Zydus Cadila, and Moderna vaccines are authorized in India and are being produced by various different companies. Now I'm going to move into the therapeutics. So far we have discussed about the vaccines, their development and their examples and approved vaccines. Now I'm going to move into the therapeutics. Therapeutics are kind of useful when the infection is not stopped. Infection, body already infected virus is producing more and more. To reduce the viral infection, to cure the viral infection, we need the therapeutics. Approximately 506 potential candidates are under development, and 419 of them are in the human clinical trials. 2 to 3 of them are already authorized for emergency use. One of them is for the completely approved for the use in COVID-19 infected patients. The very first drug molecule is remdesivir. This is the first and only antiviral treatment approved by FDA, completely approved by FDA to treat COVID-19 adults and pediatric patients as well, like 12 years of age above. Moderate to severe, so who has a moderate to severe symptoms. And this is the structure of the remdesivir. Actually, it is a repurposed drug developed by Gilead Pharmaceuticals. In 2016, this drug they developed against the Ebola virus infections. And then during the development process, they also tested this against other coronaviruses, SARS coronavirus 1 and MERS coronavirus, which as an epidemic, they came in 2002 and 2012. And then as soon as this COVID-19 pandemic started, they repurposed the same remdesivir drug molecule against the SARS coronavirus 2. And then they found a very good activity. And sometime in May, they authorized for emergency use and then fully approved in December 2020. So this is the structure of the drug, which resembles adenosine DNA base nucleotide, which builds human DNA. And this one goes through, once it is injected inside the body, goes through several enzymatic reactions and then forms this triphosphate, remdesivir triphosphate form, which is the active form of the drug. Here is the synthetic scheme that I provided here. Since the chemistry students, I'm presenting this seminar to the Department of Chemistry students. So I thought maybe, it would be nice to show some chemical structures, chemical synthesis here. So this one has about 15 to 20 steps. Here you can see there are multiple intermediates need to be produced. First, this intermediate, and you can see that they are protecting the amine group here in this kind of amino acid, protected amino acid to produce this intermediate. And the sugar was, protected sugar was oxidized to the ketones, then which is brominated. The other intermediate, this is the second intermediate. The third intermediate is this heterocycle, which is the base, brominated in this step. Then amine protector, and continued B and the C together to generate this kind of nucleoside. And then once the nucleoside generated, this hydroxyl group is converted into cyanonitrile, which is a cyanide group, using these two steps. And after that, the alcohols on the sugar moieties are deprotected. Then these steps, after these two steps, the molecule, the remdesivir molecule, it's a diastereomeric mixture of will be produced, and then chiral, based by chiral resolution, the pure remdesivir will be synthesized, made. These are the steps here. So the way the remdesivir acts in the human body once it enters. So when I say, the virus infects the body, it starts producing the more and more virus using an enzyme called RNA-dependent RNA polymerase. So this remdesivir inhibits the activity of RNA-dependent RNA polymerase. So let's say this is the virus genome, virus RNA. So RNA-dependent RNA polymerase is producing more and making a copy of this RNA genome. So the remdesivir will go inside and incorporates into the newly synthesized genome, and then it stops the activity of the RDRP enzyme. Here is the cryo-electron microscopy structure of this remdesivir molecule bound to the enzyme binding pocket. This is the virus genome RNA, and this is the newly synthesizing RNA. So this is how it blocks the enzyme activity and then stops the virus production. So that's how it cures the viral infection. So recent clinical data suggests that it's collected about 562 participants participated in this clinical trial with 279. This is a double-blinded, it's placebo-controlled randomized study. 279 people in the vaccine group, 283 people in the placebo group. So only 2 people got the infection among these 300 people, 279. But in the placebo group, placebo means these patients were not given the remdesivir. These patients were given the remdesivir only. About 15 patients were detected with the virus infection, indicating that it's still remdesivir is 80% reducing the risk. 87% of the risk is reduced. COVID-related hospitalization risk when they're treated at the appropriate time. And this is the clinical data. You can see the number of cases increased in the placebo compared to the remdesivir. And these are the brand names and the companies that are producing. This is in the United States and this is in India. And some other therapeutics are also authorized for the emergency use. One of them is Virafin by Zydus Cadila. This is an immunomodulator. It's a pegylated interferon alpha 2b, also utilized for the authorized by DCGI for moderate to COVID-19 infections in adults. And there is a combination therapy together with the remdesivir, another immunomodulator called baricitinib, which is the Oluminant, its brand name, which is standardly used for the rheumatoid arthritis. Also authorized for the emergency use by FDA for hospitalized patients with a suspected laboratory confirmed COVID-19 infection. And some antibody therapeutics also have been authorized for the emergency use. From Eli Lilly, this bamlanivimab monoclonal antibody. These antibodies are isolated from the human's sera and purified, produced, synthesized in the lab, and purified and produced and utilized as the potential therapeutic. An antibody cocktail, 2 antibodies, 2 monoclonal antibodies together, cocktail from the Eli Lilly, and the RegenCov, 2 antibodies, monoclonal antibodies, casirivimab and indevimab, together as a cocktail from Regeneron Therapeutics. They have authorized for the emergency use. So, however, we have produced, we have discussed about the vaccine development, therapeutics development, and lots of people, as soon as we got the vaccines authorized in December 2020, thought that the pandemic is going to over within months or maybe in a year. But the virus started mutating and started developing the variants, and then it started fighting against the vaccines that we have developed and the therapeutics that we have developed. So what kind of mutations the virus producing? So the most important part in the virus developing the mutations is the spike protein. So this is the cryo-electron microscopy structure of the spike protein, and this is a 2D map, which has 2 subunits, S1 subunit and S2 subunit. It can be divided into parts, and this is a site where the protease can cleave. And the most important part is this is called receptor binding domain. In the receptor binding domain, this orange part is called receptor binding motif. You can see that the amino acids 438 to 506, these amino acids play a very crucial role in binding to the ACE2 receptor in the human body so that it can infect. This is a receptor binding motif. And these are the cryo-electron microscopy structures of the 3D models. Only spike protein, different views. This is together with the ACE2 receptor. So the first mutation started with D614G. At this particular place in the spike protein between the junction of the S1 and S2 subunit, which is started in sometime March 2020. And D614G is basically, this is the original wild type. The Wuhan virus has this sequence amino acid. This is the at 614th position with this nucleotide sequence. These 3 nucleotides represents the aspartic acid, which can be said a D in the short form. When one nucleotide adenine changed into the guanine here, and this 3 nucleotides represents the glycine now. So that's how D614G the name got. So the aspartic acid mutation changed into the glycine because of this single change, this single nucleotide change. So this started in March 2020, and it has a high infection rate. By May 2020, 70% of the cases all over the world are detected by this particular D614G variant. In December 2020, within about through 3 or 4 months, virus started mutating even rapidly. Alpha variant produced with the additional mutation in the receptor binding motif. You can see that N501Y in this particular region here, which has much more 50 to 70% more infectious than the D614G mutant. However, it doesn't show any kind of impact on the monoclonal antibody treatment, and it showed a minor impact on the vaccine that we produced. So it was not able to spread out all over the world, but much more rapidly. It has about 7 mutations in the spike proteins. The next variant in the same December 2020 in the South Africa, the alpha variant was developed in United Kingdom, but this beta variant was appeared in South Africa. This one has 9 mutations, more than alpha variant mutations. Now you can see that within this receptor binding motif, there are 3 mutations in addition to the mutation that we saw in the alpha variant, as well as the D614G. It significantly impacted the antibody therapeutics, monoclonal antibody therapeutics. Some of the antibodies are not working against this variant, and moderate impact it showed on some of the vaccines, like the efficiency is reduced. And in January 2021 in Brazil, gamma variant appeared, which has more mutations than the other 2 variants. About in the spike protein only, we're talking about the spike protein here, 12 mutations in the spike proteins, and it has 3 additional mutations compared to the beta variant. There is a change in the mutation, but still it has 3 mutations, but there are other mutations accumulated in the spike protein, including the mutations that we saw in the beta variant, which showed a significant impact on the antibody therapeutics and a moderate impact on the vaccine efficiency. And now, in April or May 2021, another devastating variant, Delta variant, showed up in India. This has, again, only 3 mutations in the receptor binding domain, but they are different from the other 3 variants. But it also contained 2 mutations, one from the alpha, beta, gamma, which is N501Y, and D614G from the original first variant. It has some other mutations in all over the spike protein, approximately 17 mutations. I listed only a few of them in here, but this variant was highly infectious, more 97% more infectious than the other 3 or 4 variants. It showed potential impact. Some of the monoclonal antibodies are not at all working against this Delta variant. And potential impact, the efficiency went really drastically down of the vaccines with this Delta variant. Now, in November 2021, the Omicron variant showed up. So this Omicron variant has approximately 15 mutations in the receptor binding domain and more than 3 to 5 mutations in the receptor binding motif, and approximately 37 mutations in the spike protein. This is showed up in Botswana, South Africa in November 2021, which is much more highly infectious than even Delta variant, much more faster. And this showed very significant impact on the monoclonal antibody treatment, but somewhat smaller impact on the vaccine efficiency. It's a milder infectious. The severity is lower, but infection rate is much faster than the Delta variant. So here in this chart, what I try to explain here is the efficiency, how the variants impacted the efficiency of the vaccines. You can see that the Covaxin, Bharat Biotech's vaccine, all of them showed, but it maintained the Delta variant till the Delta variant. They lower the efficiency, but still the vaccines are working fine. Sinovac as well as same. Some of them are not available. Data is not available, but Oxford AstraZeneca vaccine, if you look at this one, the beta variant significantly impacted the Covishield Oxford AstraZeneca vaccine, but the Delta variant retained its efficiency. So Johnson, overall, all the vaccines are retained their efficiency, but we do not know how well they are acting against the Omicron. So the question is, why vaccine efficiency is being reduced? Does that mean the vaccines are not going to work anymore? Do we need new vaccines? The current vaccines are going to be no more useful? These questions raised. So let's take a look at the answer. So the vaccine efficiency influence various different factors. The first thing is host factor, which is the old age, previous infections, immunocompromised patients, various different host factors, right? Human nature, the body nature, demographic factors. So some of the region, like in South Africa, they have some of the people has immunocompromised patients, things like that. Vaccine accessing factors like availability, and the vaccine variant factors, again the one that we have discussed in previous slides. The more the variant, the more mutations in the variants, the less efficacy is going to be the vaccine. The other the factor is the immune factors, the antibody titers, how long the antibodies are retaining in the body, how well they're fighting with the infection. So usually when the vaccine is given or the person is infected with the potential virus, the body develops various different types of antibodies. I gave only the antibodies here as an example, antibodies that are developed against this particular spike protein. If you see that there are various different parts of the spike protein, right? So internal antibodies, these are the one type of antibodies developed. Receptor binding domains, certain antibodies that only target the receptor binding domain, certain antibodies that are developed to target the S2 subunit and other parts of the spike protein. And moreover, the most important point here is the spike protein, the receptor binding, the antibodies that are developed against the receptor binding domain. There are 4 different classes. That means, again, there is a difference between those antibodies as well. So if there is a mutations in this spike protein, let's say the receptor binding domain, that means 1 or 2 classes may not work, but the other classes are going to work fine. That's how, if 2 classes are not working, let's say 2 classes are not working, that means the vaccine efficiency is going to go down, because 100 out of the 100 antibodies are not fighting efficiently against the infection. Let's say only 90 out of the 100 are working efficiently to fight against the infection. So the antibodies also, how they work, they go and bind to the spike protein, so that the spike protein is not going to go and bind to the ACE2 receptor and it's not going to infect. And some of the antibodies also going to go and bind to the ACE2 receptor, so that the virus, they're not open. The ACE2 receptors are not open for the virus spike protein to bind, so that it will infect the cell. So recently, everywhere, all over the world, people are talking about the booster doses. How does these booster doses help? Here is the data, clinical data. So this is a clinical data recently published. It's a small sample size, about 51 participants were used. So what they found, how this study was done is, so the human cells which produces the ACE2 receptors will be cultured in the lab, and then the vaccine serum collected from the person who received the booster dose and mixed with the pseudovirus, the virus that has the spike proteins. It's not the real virus that has the spike proteins, which has a receptor reporter molecule. It infects the human cell, it glows with some kind of a reporter. If it doesn't infect the virus, human cell, it will not glow. That means it's successfully neutralized. So they together, the mix of human sera and virus, the virus will be infected to the human cells. If it glows, it will show the signal, and then you can read it through the spectrophotometers and things like that. So here is the data for Pfizer BioNTech vaccine, COVID-19 vaccine, after the booster dose, for various different variants. The Wuhan variant is the old wild type variant, Omicron, Beta variant, and Delta variant. You can see that. Wuhan variant, after the 2 doses, significant antibodies are still produced, but Omicron, you can see that the antibodies are not successfully neutralizing the Omicron variant that are produced for by the 2 doses. And even Beta variant, they're not, like I said, not all the antibodies are neutralizing the virus. Only a few of them. That's why still most of the cells are being infected. The other one is the Delta is even worse here. You can see that, right? So after the booster dose, the antibody, the neutralization titers went all the way up for all the variants. You can see where after second dose. So I think this data collected after 3 to 4 weeks after the booster dose. It still retains for the original wild type, the neutralizing antibody titers. the increase, there is a 23-fold increase for the Omicron variant compared to the 2-dose versus 3-dose. Similarly, there is a double increase for the Delta, for the Beta virus, and then a mild increase for against the Delta variant. And also, this is additional data. This for the other vaccines, mRNA 1273, Moderna vaccine. This is again the Pfizer vaccine, Johnson & Johnson vaccine. You can see that for the wild virus, still after the third booster dose, you can see increasing levels of neutralizing titers. For the Delta, about 16-fold increase with the Johnson & Johnson vaccine after the third dose, from about 9-fold increase for the Pfizer-BioNTech vaccine, Moderna vaccine 3-fold increase, Omicron variant 4-fold increase, 27-fold increase by Pfizer-BioNTech vaccine, Moderna vaccine 19-fold increase, Johnson & Johnson vaccine 4-fold increase after the third booster dose. So the data shows that the booster dose really helps. It produces more antibodies to successfully neutralize the infection. And this is how there's another data point that I'm presenting here is, so the vaccines after the prior infection, let's say after 2 doses, somebody get the COVID-19 breakthrough infection, COVID-19 infection. How is their antibody titers? They're also equally producing the antibody titers equal to the 3 booster doses. So that means they're also producing very well. Various different variants are tested. You can see Delta, Omicron. This is the booster dose level antibodies for the different variants, wild type, Delta, and Omicron. This is a person who had the infection after the 2 doses. This is a breakthrough infection. And this is with the Pfizer-BioNTech vaccine, Moderna vaccine comparison with the Johnson & Johnson vaccine here. And there's another good thing here is some of the companies started developing new vaccines as well. So Moderna, for example, started developing variant-specific monovalent vaccines. So the original mRNA 1273 is the vaccine that is developed using the spike protein genome information of the wild type original Wuhan virus spike protein genome. But now they are developing mRNA 1273 351. Again, they're using the beta variant spike protein. This is a Delta, sorry, I put the beta, but this is a Delta variant spike protein. This is another monovalent vaccine. They're also developing multivalent vaccines. So the D614G variant, beta variant spike proteins together in one vaccine so that it will successfully produce 1:1 mixture of the 2 variant spike proteins. And this is 1273-213, mRNA 1273-213, beta and Delta variants spike proteins together in 1:1 mixture. They have very good results here. Produced the clinical data here, the antibody neutralizing titers against Omicron variant. This is particularly against the Omicron variant. Original mRNA 1273 50 microgram dose, 100 microgram dose original vaccine that currently is being under emergency use authorization. This is a new multivalent vaccine. This one, you see the Omicron versus the wild type. You can see a very decent neutralizing antibody titers. And this is with 100 microgram dose, is more better than the 50 microgram dose. And this one is a Delta Beta multivalent vaccine against the Omicron variant. And this is how antibody titers are going down against the Omicron variant after 6 months. This is the data that shows here. So how does these vaccines influencing the hospitalizations in the USA? So if you look at this, this is the real-time data from the CDC. So the fully vaccinated persons, the hospitalizations are almost none. You can see that right with the time. This is like from June 2021 to December 2021. It's consistent. No hospitalization drastically reduced. Unvaccinated people is keep on increasing. You can see that as the wave comes in, the hospitalizations are being increased. That means vaccine really helping the people from hospitalization and severe infection hospitalization. And this is for the 12 to 17 years kids. This is between 50 to 64 years adults. You can see that fully vaccinated people is the blue line with the booster dose. This the bottom blue line is with the who got the booster dose. Hospitalizations are quite drastically reduced. This is only with only 2 doses also protecting very well. But you can see that unvaccinated people keep consistent, continuously going on high hospitalization rates with severe infections. And this is the death rate, how the vaccines are helping controlling the death rate. This is the fully vaccinated with the boosters. Death rates are almost zero. If you get the booster dose, the chances of death is zero with COVID-19 infections, pretty much. You can see that. Fully vaccinated means 2-dose vaccines, but no booster. There are still chances of getting into because these are the immunocompromised patients. They have these are not the healthy, but immunocompromised patients are people with the comorbidities. But you can see that unvaccinated people, you may think that the rate is going down. It's not going down. It's because there is no variant here at this point. There was a wave here. There's no wave, and then now the Omicron wave started increasing. It's going to go all the way up. So which means vaccines boosters are really helping from protecting the people from hospitalizations with a severe illness and as well as the death. How does the therapeutics are helping protecting the how they're working against the variants? This is a clinical trial data that I'm presenting, and they use this new test called live virus focus reduction neutralization assay, FRNT. So that means once similar to the one that I explained, the pseudovirus assay, they infect the human cells with the virus after giving the therapy, infect the virus and give the therapy, and then observe the foci. The virus forms a foci. How many foci? They count the foci. More the foci means highly infected. So if you can see that, these are the antibodies that are approved, authorized for emergency use for the initial variants like wild type, alpha, beta, gamma variants. Initially, they are working fine for all the alpha variant, beta variant. Beta variant is not very well for this particular. Gamma variant, they are fine. Delta variant also, these antibodies treatments are working fine. But Omicron variant, you can see the foci number is thousands. That means it's not controlling, it's not working anymore on the Delta variant. These authorized antibody therapeutics. There are 2 new antibody therapeutics that are on the clinical trials. They are kind of showing some promise against the Omicron variant, and in addition to all the other variants. Also, these are the drug molecules that are derivatives of the remdesivir and other molecules, showing some promising results against the Omicron and all other variants. So on January 26th, last week, I think 24th. So FDA limited the antibody therapeutics, as I mentioned in my previous slide, since these antibody cocktails as well as the monoclonal antibodies are not effectively neutralizing the Omicron variant, so they limited their use on Omicron infected patients. So this is being controlled. This is a new development in the last week. And also additionally, like I said, there's a new monoclonal antibody, sotrovimab, is being authorized for the emergency use for the Omicron infected. It's working very well, all the COVID-19 infections, and it's efficiently working against the Omicron variant. Here is there is a clinical trials data. Again, this is about 600 participants with double-blinded placebo-controlled study. In this sotrovimab group, only 3 people got the infection, but the placebo group, 21 people got the infection. So it's still a highly protecting. And these antibody therapeutics are also prescribed to high-risk patients only who are not hospitalized and with a positive COVID-19 test. And this is approved for the adults as well as the children above 12 years old, at least who has the 40 kilograms of weight. Another new therapy was approved recently a month ago by FDA, which is a molnupiravir. This vaccine also not repurposed. This therapy also repurposed. This is the structure of the molnupiravir, which is called EIDD 2801, developed by Emory University originally, and Ridgeback Bio and Merck together, they commercialized this molecule. It started against a different virus, EE virus infections in 2013. And in the development process, it is repurposed. When it is repurposed in May 2020, April 2020, it showed some promising results, and then it went through all the clinical trials and authorized for the emergency use in 2021. So this is the original structure, which resembles the uridine, another nucleoside, which converted to this form in the plasma, then other enzyme converts into the triphosphate form, which is the active form of the drug molecule. This one also inhibit the activity of the RNA-dependent RNA polymerase, which replicates the RNA of the virus. What it does is introduces more mutations in the virus genome. Instead of the regular ATGC bases, the molnupiravir goes inside them and then deactivates the virus RNA so that the virus will not reproduce more and more. This is how it works. And so early treatment with the molnupiravir reduces the risk of hospitalization and death in at-risk, again, highly risky patients and unvaccinated adults as well. This is also one of the potential therapy approved last month. This is the synthetic scheme of the molnupiravir. It starts from this uridine base. It is the original published patent from the Merck. It starts with the uridine. First, these alcohols are protected with the acetonylation, then esterification of the alcohol, the secondary alcohol, primary alcohol down at this position, fifth position. Then the triacyl group introduced, and which is converted in the following step into the hydroxylamine, and then the acetonide group is deprotected, and crystallization will give the pure molnupiravir molecule. Another drug molecule was approved last month, which is called Axlovite. It's a combination drug. The original molecule is Neuromatrial, which was developed by Pfizer. This is an oral pill. The molnupiravir is also an oral pill. So this was also an oral pill, but remdesivir is an injection. So Neuromatrial is the original molecule developed by Pfizer. This is not the repurposing. This is original, completely developed based on the structure-based design from this molecule, which was in the repository of the Pfizer. This inhibits the protease activity. 3L protease, one of the enzymes that helps the virus to infect the human body. Here is the molecular modeling structure where this drug molecule bound into the binding pocket of the protease enzyme. And this is given together with ritonavir. This is already approved drug for HIV infections. What it does is this molecule inhibits the protease activity, but ritonavir doesn't do anything to the virus, but it retains the Neuromatrial's activity. So it protects the Neuromatrial from other metabolic enzymes so that it will not removed by the metabolic enzymes from the body, so that its activity retains long period of time. Again, this is authorized for it showed 89% effective in patients at high risk and serious illness for serious illness patients. Here is the synthetic scheme of the Neuromatrial. It starts from this amino acid, protected amino acid. It has a lot of chiral groups in here. The first thing is so converting this ester into the amide, then amide deprotection, Govt deprotection, and then this is one of the intermediate synthesized. Then it starts from this bicyclic chiral molecule, cyclopropane ring, and then as amide chemistry, amide bond formation at this secondary amide down here, and then after this, you can clearly see that here the esterification, right? Here this acid ester reduced into the acid, then Govt deprotection again. After that, then the trifluorocarbon, again amide chemistry, peptide chemistry introduced with this moiety down here. Then using this key 13 molecule will be introduced at this particular acid group. Again, amide chemistry, acid peptide chemistry, amide bond formation. And in the last step, they use the Burgess reagent. This is a dehydrating reagent. It's commonly used to generate the alkenes type of double bond or triple bond. Basically, they use this molecule, this reagent, to generate this cyanide group at this amide, from this amide. So this is a synthetic scheme of the Neuromatrial. So far I have discussed about the coronavirus infection and immune response, various methods of vaccine development, and its structure, coronavirus structure, mutations and variants, authorized approved vaccines for the COVID-19 infections, and authorized approved therapeutics. Authorized means emergency use authorized therapeutics for the COVID-19 infections, latest developments in the vaccines as well as the therapeutics. And then I also showed them how the therapeutics and vaccines and booster shots are helping controlling these deaths and hospitalizations so that we can come over with this pandemic. And the one last slide I would like to say here is, even though we have vaccines, therapeutics, everything, how to protect ourselves from the COVID-19 infections. There is the 3-number rule. The very first thing is trust the facts and get the vaccine. Pharmaceutical interventions, which is whenever is vaccine available or whomever is it available, go and get the vaccine, number 1. Number 2 is living a healthy lifestyle to boost your immune system, eating healthy food, getting sufficient sleep, and being inactive, doing some exercises. And the third one is non-pharmaceutical interventions. There are 5 things in here. We have been talking for past 2 years, but the first 3, I said that these are very important, regardless of your vaccination status, your infection status, whatever it is. Using the mask until the pandemic ends protects you from the infection. Frequent hand washes protects you. Sanitization when you go outside of the house protects you. And you can, we can relax a little bit on practicing social distancing and avoiding social gathering. These 2 other non-pharmaceutical interventions, if you are fully vaccinated or if you got the booster vaccine, and you can relax these if you wear the mask. That's what I would like to say. And having said that, in this my journey started from Department of Chemistry, Usmania University campus, where I started learning chemistry and learning about the live chemistry. Campus gave me a lot. Then after that, I joined IICT, where my research career started. Where I set up my first chemistry reaction towards the research. Then I moved to University of South Carolina, where I did my PhD in chemistry. After that, I went to here I did purely organic synthesis and made some material science applications. Then I moved to the Wayne State University as my postdoctoral studies, where I did some medicinal chemistry over here. I developed some antiviral, antibacterial novel agents. I learned how to culture the bacteria, how to test the medicinal compounds against the virus and bacteria. Then I moved to MIT as a research scientist, where I started working with the human cells, studying the biosynthetic pathways of cancers in human cells, while using the skills that I've learned in all these different places. Right now, I'm working at Expansion Technologies as a head of the research. We are developing novel early stage diagnostic methods for various different cancers. Having said that, thank you so much for your attention. My apologies, I took a long time. I am more than the required time, about like 10-15 minutes more. But thank you so much for your attention. I'm happy to answer. But last but not least, I have to thank all these people. Lots of people, my students, my colleagues, my gurus, my friends, my family members, everybody helped during this journey. Without their help, I don't think what I am today and where I am today. Having said that, thank you so much for your attention. I'm happy to take any questions if you have.
Thank you so much, sir, for your elaborative discussion. I request all the participants to kindly involve in the interaction or interact with the resource person. There are some questions, sir, in the chat box.
Sure. Sir,
the Neovac virus, that is Neocow virus, how is it going to attack? Is it same as much virulent as that of Corona? COVID-19.
A good question. Yeah, nowadays, in the news media channels, you see a lot of this, right? So we don't know much about that new virus unless until these news channels and everything, right? Studies are ongoing right now. I would not worry about that. Like, it may not happen like the COVID-19, but we should not take the chance. So that's why, again, since we are already going through a pandemic, what I would like to say is, don't worry about these kind of new viruses. There are so many viruses that are already there in the world. Always there are new viruses going to come in. When there is an Ebola virus pandemic there, it just stayed to a certain region in South Africa. When there was a SARS coronavirus 1 epidemic was there in 2002, it was only limited to certain number of countries. There was a MERS coronavirus infections epidemic in 2012. It was also limited to a certain number of countries. So it all depends on the situation of the virus and conditions, and then demographics, like I mentioned, various different factors involved. So right now, I wouldn't worry about it virus, but I would.
Mahender sir, please kindly unmute yourself.
Hi. Did I answer the question or did I?
Last minute it was muted, sir. Last.
Oh, okay. So what I would like to say here is, there are a lot of viruses already there in the world. New viruses always are going to come out, but we do not have to worry about the new viruses coming in. They may not be highly infectious. There may not be there may not be another pandemic, because there are several epidemics were there. It may restrict it to certain region. So I wouldn't worry about much about this new virus that is being shown in the news channels, but I would be cautious, happy. I would say I would take precautionary measures, like right now we are already going through a pandemic. Use non-pharmaceutical interventions to protect yourself, like use the mask, unless as long as this pandemic goes on, until the pandemic ends, and continue taking the vaccines wherever it is necessary, whoever is eligible, whoever has the vaccine available. That's what I would like to say. So, but the neo virus, not to worry about too much, because we do not know much about it. We do not know how infectious. Maybe in a couple of weeks we will have a better idea.
Okay, sir. A few more questions are there from the participants.
Please.
Yeah.
Go ahead, no problem. And anybody has a question, they can unmute themselves and they can ask a question as well.
Yes, sir. I'll allow the participants also to do that. So before that, I'd like to ask a few questions, which I have collected from the chat box.
Please go ahead.
Chances of herd immunity with fast changing virus mutations and protection from infection in future. How is this going to be like?
Excellent question. The herd immunity is a theoretical concept. What it says is about the community, about the people, out of the 100% of the people, 75 to 80% of the people either gains immunity through the vaccine or through the prior infection. They will have a sustained immune system, sustained immunity against the virus infection, so that less people will be infected. That's how virus will go away from the society. That's the concept, right? There is a possibility, now you can clearly see that the vaccines, as the vaccination rate is going on and up and up, the infection rate is, the hospitalization, the death rate is going down. That means the severity of the vaccine virus is kind of already being controlled, right? The infectious rate is being increased. More people are being increased, even though they're vaccinated, like breakthrough cases are coming. It is because of the variants. And that variant also is going to go away when the virus mutates more and more and more, because the mutations is a virus's natural instinct. It will mutate all the time. As long as the human body is available to the virus, it will start mutating. And the mutations also, it could be bad mutations, or it could be good mutations, in the sense of bad and good for the virus. Where there's a good mutation, variants like Omicron, Delta will come out. When there is a bad mutation, the other variants, the suppressing variants are going to come out, and then the virus will go away from the system. So there's a method. We are introducing pressure on the virus using the vaccines, using the masks to reduce the infection, right? Other non-pharmaceutical and pharmaceutical interventions. But virus will fight against us to produce more and more effective variant, right? So in this fight, so if we don't provide the host body, if we can't find the host body, the virus can't find the host body, what it does is it go and find animal body. Another animal go and become silent. So we'll go away from the society. That's how it will be vanquished. If the virus finds the human host continuously, it will keep on evolve and continuously make the more and more variants. And then we have to, again, continue to fight it. So what I would like to say in the end is the herd immunity will come when 75 to 85% of the population has virus protection immunity. But how that happens, it varies, because we are getting the immunity from the vaccines, but after 6 to 8 months, the antibody protection is being waning, right? So that means can we take that as a can we consider still them into the herd immunity? Don't know, not sure, right? So it's a mix and match. So I wouldn't trust 100% on herd immunity, but it's a hopeful situation, I would say. Again, still, only herd immunity is not going to get away with the pandemic. We need a vaccine-induced immunity is also very important. As the vaccine rate increases, in addition to the natural infections, once the vaccine vaccination number also goes up to 75 to 85% of the population, then that point we can say that herd immunity is there, and then virus is going to go down.
Okay. So there is another question. How vaccines affect the immune system, and does it suppress immune system to other viruses as well?
No, like I clearly explained, right? Every vaccine develops the immunity for that particular virus, because we are using that particular virus's properties, our virus itself, our virus protein, our virus genome, to produce the immunity in the body, right? To activate the immune system. So the antibodies produced only recognize that particular virus, and the memory T cells and memory B cells produced only recognizes that particular virus. Let's say if you are given a vaccine of polio virus, so the immune system activated, the antibodies produced, the memory cells produced are going to act against the polio virus, not against the other viruses.
So, sir, Professor Ashok sir would like to ask something.
Please, sir, go ahead.
Mahender ji, what about 2-deoxyglucose? Is it effective?
So, yeah, it's a very interesting question, sir. So the 2-deoxyglucose, what it is doing is very indirect mechanism. Somehow, very basic, 200 to 300 patients they tested somewhere in a hospital in Delhi. And after that, we don't really see much data on that thing. But what it is happening is indirectly. So when virus infects the human body, virus is going into the lungs, right?
Okay.
So lungs, when virus started producing more and more in the lungs, and immune system try to eradicate the virus, and it introduces more and more cytokines and chemokines, which are the inflammatory markers. And these cytokines and chemokines are supposed to go and kill the virus. But what virus is escaping, and then these cytokines are attacking the lung cells, damaging lung cells, lung tissue damaging. Then people started getting breathing problems, right?
Yes.
When it is breathing problem starting, that means they are trying to get more oxygen supply. Oxygen supply is basically reduced in the lungs, and then we need more oxygen supply. That's why ventilators, people are giving more oxygen because lungs are damaged. They're not taking enough oxygen to supply to the blood, right?
Yes.
So what this TX-glucose does is it basically starting material for enzymes that utilizes lots of oxygen to produce the metabolites that body uses. It's called hexokinase enzyme. So it's a very indirect mechanism. So what 2-deoxyglucose maybe doing is it reducing the oxygen stress on the patient.
Okay.
So that's how it probably controlling the serious illness. But other than that, it does nothing to the virus. It's not really acting on a virus at all.
Okay.
It's not like a molnupiravir, not like a remdesivir. It's not directly on virus infection mechanism, or not on the virus replication mechanism. So it's very indirect mechanism. There's not a lot of studies available either.
Okay, thank you very much, Dr. Mahender ji.
My pleasure.
Nice explanation.
Thank you, sir. Thank you.
There is Sridhar B. There's one of the participants would like to ask a question.
Please.
Sridhar sir, could you please come
Very good morning, sir, Mahender sir, and to all of the distinguished speakers. Sir, interestingly, that we need to know about the subject related, I'm asking. You have said the microRNAs are being using more interesting in therapeutics. So how could I specifically select the this main RNA could be more useful in this kind of therapeutic studies? Because I do not aware how could I select the microRNAs for like 132 and 123, like LSD1, it could be more effectively working. So in these conditions, how could I specifically select what kind of the microRNA can be able to selectively can be potential effective, I can show that giving any more or 2 or 3 microRNAs together can be functionally effective on this kind of viral infections.
Great question, Mr.
Yes.
Nice question. So,
yes.
siRNAs, miRNAs are
Yes.
still under the development. As of my knowledge, I don't see any therapy yet approved or authorized for any kind of treatment yet. Still under development. The RNA vaccines situation 20 years ago is the same. 10 years ago is the same. So now, the way that there are various approaches. I'm not an expert in that microRNA or siRNA field, but there are various different approaches. For example, RNA therapeutics, it took like 3 or 4 decades to come into the therapeutic, use, particularly with this mRNA-based vaccines, right? People tried so many different things. The major problem for the mRNA therapeutics is the delivery methods, right? Because RNA, as soon as the RNA is discovered by the enzymes in the body, they're called nucleases. What they do is they clear out the RNA. They kind of chop it off, they delete out. So that's the protection is most important rather than the selection. So I think that's what is probably causing the microRNA also reaching to the target. Also one of the important part, right? You're delivering into the body, but where you're delivering, where it is going. Like let's say cancer, is it going there or not? That's another problem. But selection can be done on multiple factors, not for one factor. So I'm not an expert in that field. I cannot give you like exactly straight answer, but I'm happy to discuss with you offline if you have any. If you would like to discuss further in future, please feel free to reach out to me.
Definitely. I'll try to do that one. Thank you.
Yes, please go on, sir. Sridhar, sir, please go on.
As you said, the microRNAs, it will never directly reach to as far as my concern, because when the previous 3 years back when I was studying these microRNAs which are been using, they are site specifically they are introducing. Like some of one patient will have the P101 mutation for having this insulin resistance for this sugar patient, diabetic mellitus. So for them, they are releasing on site specific in their gene itself. So to modify like microRNA 132 and 123. These two microRNAs are again they are using in these eye diseases and all so many are those things. So why I'm interested in this? So many factors are there by using these microRNAs, but how could we able to select? There is no specific answer so far. I have found it so far. So is there any databases there that I could more dependently to get those kind of answers? And that
let me look into it because I don't have a right answer on top of my head at the moment, since I am not an expert in the field.
Fine.
But I'm happy to look into it and get back to you. Please feel free to reach out to me offline.
Thank you.
Yes, sir. Sir, there is a question from Dr. D. Narmada. What should be the gap between second dose and the booster dose? Is it exactly the 9 months period?
It's a very good question. See, it all depends on, again, multiple factors, right? So, for example, let's say mRNA-based vaccines. I have showed some data that, for Pfizer-BioNTech vaccine, the antibodies are retained for up to 8 months. Active and retained in the body up to 8 months. For Moderna vaccine, only up to 6 months. So it varies. It depends on the vaccine type, number one, and also vaccine availability. And based on multiple factors, the authorizations, they're deciding this 9 months or 10 months or 12 months. But in the United States, FDA authorized a booster dose after 5 months of the second dose. It's because the vaccines are available. And we have a data showing that after 6 months, the antibody titer is going down. So that's why they made a decision of after 5 months. So 9 months in India is probably the vaccine availability, right? We have a very heavily populated country, and then we only have like 4 or 5 vaccines authorized, which may not be sufficient amount produced to overcome the need, right? So probably that's based on that particular. I think the 9 months is based on the availability. I believe. I'm not sure. I don't know. So like I said, it all depends on the data. I do not have much data available I do not have much data on the Covaxin. The Covishield also waning about 6 to 8 months. So pretty much all the vaccines, pretty much 6 to 8 months in general, that range. Their antibody support is going down. So, I mean, I would say, whenever it is available, go and get it. Don't worry about like this month, that month.
Okay. So I request all the participants, if anybody is there to like to ask the questions, please you can raise your hands or you can unmute yourself and ask the questions.
Sir, why the vaccines are not developed for HIV and HPV virus are rampant, similar to COVID-19? What is the reason?
Yes, there could be many reasons. Like right now, only there are a couple of methods available until this COVID-19 pandemic. Now we have a new platform, complete new platform, which is a next generation platform using the DNA or RNA based vaccines. Right before that, we only have the conventional and classical methods. So that's one of the reasons, not a lot of platforms, excuse me. And HIV infects the immune cells itself, T cells. So HIV is a very complex virus. It's a very complicated virus. And as soon as there are multiple therapies already developed, and within a few months to years of period of time, it develops the resistance to those therapies. So that's why the HIV is a pretty complicated virus. So that's why it's taking a long time. But I'm hoping that with this RNA-based vaccine development method, I'm hoping that soon we're going to get the vaccines for HIV.
So Narasimha Rao Garu would like to ask some questions. Sir, please you can unmute yourself and you can ask. Narasimha Rao Talisetti Garu.
I think he is muted.
Been given co-host. Okay, so if okay, Dayanand sir.
Sir, I'm having one question, sir.
Yeah.
Hello.
Hi.
Sir, what are the negative effects of this vaccine, sir? Already you have said there are so many positive things. What are the observations by you? Because the most of the people they are blindly taking, but maybe it may be having some adverse effects also. Have you noticed anything like that?
Yeah, so let me explain that. It's a great question. So all these vaccines, whatever the vaccines that are authorized all over the world, approximately 11 to 12, they've underwent a severe clinical trial process, phase 1, phase 2, phase 3, before the preclinical development, clinical, multiple, and also in the phase 3 clinical trial, they went through 30 to 40,000 of people. Even Covaxin in India, 26,000 people they tested in the phase 3 trials. So whenever you're running the phase clinical trials, they look for two things. One is systemic reactions and serious adverse events. These are the two conditions they look for when they're conducting the clinical trials. Systemic reactions means, so common reactions like, when pain on the hand, when you get the injection on the hand, obviously 1 or 2 days you will feel the pain. That's one of the systemic reactions, right? So when the vaccine is given, sometimes some people feel the feverish and chills and some mild nausea kind of things, mild fever as well. These are all the signs of activation of the immune system. So when you're giving the vaccine, means you're giving some kind of dummy virus into the body, and which recognized by the immune system as a virus, and then it will produce lots and lots. It will activate the immune system immediately, right? And then those signs will be produced outside the body like this. Some people experience this, some people may not experience this. These are the systemic reactions. Number one, these are so serious adverse events. And the second part, right? Serious adverse events means if something happens to the kidneys, brain, liver, heart, any kind of situation, if it happens, they observe during these clinical trials. So all of these vaccines, very mild, very low number of. So there is some kind of for mRNA-based vaccines, this anaphylaxis issue came out back in the days. 1 in 1 million, one case in 1 million they observe, which is very negligible, right? But so also in addition to these two risk benefit analysis, they do that. Let's say a person, the chances of getting severe disease and death by COVID-19 infection versus getting the same conditions with the vaccine adverse event. What are the chances? They estimate the risk benefit. Where will be the risk? Where will be the benefit? Obviously, there is a benefit with the vaccine, right? Vaccine is protecting. So that's how during the clinical trials, they observe all these adverse events, systemic reactions, and based on that, they make a decision. If there is a lot of, like, let's say 100 people are participating in the trial, one person experiencing the adverse event, and they estimate based on the adverse event. Usually, if it is really serious, then the clinical trial will be stopped, and then the vaccine or the drug will be stopped at that point. It will not go further anymore, right? Let's say 10, 20 people are experiencing the severe adverse events, and then it will be stopped again, right? So all these vaccine trials, the most of the adverse most of the reactions are systemic reactions, like I explained, not the severe adverse events. But some of the real-time data showed some pericarditis in the children, some of the anaphylaxis problem in the adults, and some of the blood clotting problems. It's a very rare event. It all depends on the human body. I mean, that person's underlying conditions, comorbidities. So various things will involve in there. But for healthy individuals, I wouldn't see any kind of a serious adverse events. I hope I answered the question, Dr. Shukla.
Sir, one thing I would like to ask you.
Yes, sir. Thank you very much, sir.
Sir, very good session, sir. Congratulations, Mahender, sir. You have given a very good information. And one thing I would like to ask you, sir, whether the mix of vaccines are better or the same vaccines are better for the antibody production?
Excellent question. This is a wonderful question. I wanted to cover that part also in my talk, but I haven't had enough time to put together. So you asked a very good question. So see, antibody production, always, these vaccines are developed using the different methods, different styles, right? So recent data, there's a lot of data is coming out that, mix and match is giving better protection, better antibody. The difference is very minor, I would say. 10%, 20% difference, I wouldn't call it as a difference. Let's say same vaccine, first dose, second dose is giving 50% of antibodies, but mix and match is giving 90%. That is called the difference. I don't see that difference. So right now, and most important thing is also you need to look into the safety, how safe mix and match vaccines is, right? So we have a lot of data for especially mix and match between adenoviral vector vaccines like Oxford, AstraZeneca, Johnson & Johnson, Sputnik vaccines with the mRNA vaccines, which said is safe and produce a little bit more effective antibody production compared to the homogeneous vaccines, like same vaccine. But I wouldn't be thinking that it's really greatly producing a lot of antibodies and then a lot of good thing happening. It's same. There's a minor difference, I would call it. I hope I answered your question.
Thank you, sir. Thank you, sir.
Sir, I have one question.
This
is regarding the systemic reactions which you have referred just now.
Sure.
After taking vaccine, a few people are getting fever and some people are not getting fever or they're not showing any type of symptoms. Does it mean that the people who are getting fever, their immune system is stronger than the people who are not getting fever or not showing any reaction?
Not at all. It all depends on their own body. See, for example, even the cold, fever, flu, in the flu season, some people doesn't even show symptoms. The body goes in, and the virus goes inside their body, and then the immune system silently clears them out. That's how it will be effectively producing the body antibodies and the memory cells and clearing the infection without even showing the symptoms. Some people have some kind of strength, like their healthy lifestyle. It could be in their healthy lifestyle or their genetic, signature. So many things involved in that. But I don't think who got the fever has the more antibodies. Who doesn't have the fever or any systemic reactions doesn't have any antibodies, doesn't have any protection. That's not a measure. And also, lots of people are going for a test How many antibodies for my vaccine for this in my body? That's also wasting of time and wasting of money. It all depends on each individual. Whatever the data we are looking is cumulative, average, or data that we are looking among various different types of people, age groups, demographics, like in different countries from various different countries. So we're looking overall data. So I wouldn't take an individual. I hope I answered your questions.
Thank you.
Now I request Dr. D. Ashok, Professor D. Ashok Garu to give his remarks on this session. Ashok, sir. Meanwhile, Ashok, sir.
Yes, sir.
Please give
The
the
talk
remarks.
by Dr. Mahender Dewal is excellent, highly informative, very useful to all the category of people. I can say society is very much benefited by his talk, and I heard more than 15-20 lectures during this pandemic. Every lecture is different and very useful, and always he delivers very informative and immediately useful, immediately required talk. And even is a talk and discussion in TV also. Really, he is doing a lot in society, especially Indian society. Thank you very much, Dr. Mahender Dewalji. I am very proud of you.
My pleasure, sir. Thank you,
As
sir.
you are my own student, really, I am the most happiest person among all the participants today. Thank you so much for your excellent knowledge and excellent way of teaching. You are not only a scientist, you are a great teacher, great orator. Thank you.
Thank you so much.
Very much.
Thank you very much, sir.
Thank you, Ashok.
All the very best. I wish that you should give many more, and you have to help society in future also.
Certainly, sir. It's my pleasure. Thank you so much.
Giving me an opportunity.
Thank you, Ashok, sir. Now
Thank you.
I request Sridhar B. that raised his hand once again to give his remarks.
So thank you, sir, for giving me this opportunity. And it was a frequently meeting with Mahender Dewalji, sir, and along with our OU chemistry alumni. And thanks a lot for giving this kind of informative talk, because I more focusedly about the subject way, and so it was very informative and always gives new input thoughts in our mind, how we could be helpful to be in our society, and how could we improve ourselves and achieve in such a way that everyone can accept us and be a strong ourselves with this kind of any pandemic or any situations and so on. So it was very nice talkative and more informative and thankful, and thanks a lot for everyone. And especially for Mahender ji, sir, and organizing these kind of programs with other Kakatiya, sir, and all different universities and all. So it's really helpful, and I have a great day, and thanks a lot. Personally,
thank you so much for the comment. Thank you so much for the
Sridhar,
comment.
sir. Thank you. One more L. Tirupati is there. Kindly respond, Mr. Tirupati, sir.
Okay, sir. Thank you, sir. First of all, I greatly thankful to Mahender Singh, sir. As a non-science student, I learned a lot of things from you, sir. Really, the new terms, especially the immunity system. So I thankful for your very valid and informative session, and I also thank our organizers giving an opportunity to listen your words, sir. Sir, and my one doubt here is, sir, we are taking the first dose and second dose, sir, and there are some rules regarding the impact of those doses. My question is, in near future, not in instantly after taking the dose, is there any impact, any possibility of impact in near future, sir, on our health ground is concerned? That is my concern, my question, sir.
Sure. I can answer that question. See, Tirupati Garu, I mean, the vaccines are being given from past 2 years, right? Now, the first vaccine started in December 2020. It's now 2022, right? I think millions and millions, almost how many? 700, 800 millions probably vaccines were given all over the world, 700 millions. And if something happens with the vaccine, we could have known already by now, right? It's been almost 2 years, like you said, near future. So all these vaccines are safe, protecting very well. So I wouldn't trust any rumors. That's what I would like to say.
Okay, thank you, sir.
Okay,
now
thank
I
you,
request the senior most lecturer of our department, Dr. Eluri Yadaya Garu, to give his remarks on the session. Yadaya, sir.
Okay, Dayanand, I am audible?
Oh, audible, sir.
Okay. So, good morning, everyone. The distinguished resource person of this session, Dr. Mahender Dewal, Head Research and Development Principal Scientist, founding member, Boston, USA. I am welcoming, sir. So you've given a wonderful, elaborated, information on living with the COVID-19, role of vaccines and therapeutic to overcome the pandemic situation. It's a very elaborated and excellent session. So, sir, Mahendraji, Dewal started from birth of virus from Wuhan to how it is varies in various mutations, given a excellent and elaborated information, sir. So how the virus enter into the body and type of immunities and how vaccine protect from virus. So this information is given, and also under different type of vaccines and what is the efficiency of that vaccine, and also is given a next generation platform for develop vaccine and how it is effect on the lungs and how it is react the virus with the DNA, RNA, and given a clear and explanation with the reaction and mechanism of that variation, and also is given at the main D614G associated with the enhance of infectivity. So all the type of virus variant, alpha, beta, gamma, delta, and Omicron, impact of the variation of the variant, organization given very clear. How does booster help? New vaccine for the protection from variants, and all this information is given with a nice informative that lecture. So here you see, lastly, we given very each and everyone should be aware and should be safe zone by taking some case, and everyone wear the special mask, keep also a reasonable safe distance, wash the hands frequently, use sanitizer. By that, we will be in the safe zone and must be take the vaccine. This information given by our eminent resource person, Mahender Dewal. So here on behalf of Chemistry Department, Government City College, once again, we thankful to you, sir. You given a excellent and elaborated information, sir. This is a very, very wonderful session, we think. So thank you very much. And also I thankful to our eminent professors, Dr. Ashok Garu, Professor Ashok Garu, and Professor Veerasomaya Sir Garu. They are my teachers and gurus. So they are also participated in this session. We are very, very thankful to you, sir. And also our friends, Ramesh and Narsingh Rao and other friends, Sridhar, sir, are also involved in it and made it success. And I'm very, very thankful to from our department, from our college, on behalf of our principal. We are very, very thankful to Mahender Dewal sir. Thank you very much.
Thank you, Yadav sir. Sir, Mahender sir, can't we ingest a module so that it is potent to all viruses? It can dissolve all viruses, like new coming viruses into our body. Like the vaccines we take in the childhood, immediately after birth. Is there any possibility for such vaccine so that we can get immune against all viruses like that? Is there any chance?
So, yes. So when we're kids, we're getting some vaccines, right? They're not protecting from all viruses. The vaccines that we're getting when you're kids is the MMR vaccine, measles, mumps, and rubella vaccine. One, there is a polio virus, separate vaccine, polio vaccine, separate, and there are various different chickenpox vaccine, another vaccine. So you get multiple vaccines when you're a kid. Each vaccine is separate for each virus. But your question is very interesting and great question. I'm hoping for that in future. Any virus, one vaccine. Definitely with the technological developments, current technological developments, giving us some hope. Next, in future, near future, absolutely, we're going to see certain. Hopefully, we're going to see certain that kind of things. But for particularly coronaviruses, there are multiple companies that are developing pan-coronavirus vaccine. Let's say coronavirus 1, coronavirus 2, but coronavirus, tomorrow maybe coronavirus 3 may come. Any type of coronavirus, one vaccine. So there are companies started working on that already. So another thing also, delivery method. We are also working on nasal delivery vaccines, right? There are some companies, so that's also very important especially for the kids. So I mean, we have a hope in the future, and the work is ongoing towards that.
Thank you, sir.
And
now
a couple of other questions I do see on the chat window. I would like to address them. Okay. One question is Bhanu Rekha is asking is which kind of nutrition may help us in this pandemic, sir? So what kind of nutrition is going to help? So healthy nutrition, any nutrition. So some people are taking like vitamin tablets, great. No, it's not a good thing. You should take vitamin you can take the vitamin tablets and things like that. Like let's say 40 cross, people who are cross 40 or 50, they may take regular based, but not like crazily like vitamin D, C, all the rapidly, randomly. It's not suggested. I would recommend to talk to the doctor, prescription based, talk to them, and then based on that. And vitamin D also sometimes it's harmful too if it's too much. You should not take those type of the zinc or other things too. Some of the vitamins, essential vitamins, you can get through the fresh juices, fresh vegetables, healthy diet, and most importantly, exercise. Some of the body generates the inside the vitamins or other protection mechanisms by exercising. 30 minutes to 1 hour a day, at least walking, that certainly helps. That's what is important. Healthy lifestyle, number one, regardless of the age. Number two, the other question I do see on the chat window is from Aditya Boparaju. Why COVID vaccines are just working for 6 months, but not lifelong? It's another great question. Why not lifelong? Because let's say you take the polio vaccine one time and then it stays forever, right? You don't get the virus infection anymore. That's a great question. So the immunity development also, it's a stages. It has multiple stages. It works well for some of the vaccines. Some of the vaccines it's slow down. So when I say when you get the vaccine inside, it produces antibodies. It produces memory cells, which is memory T cells and memory B cells. Number two. So these memory T and B cells stays in the body at certain places, and when they go and settle down in the bone marrow, after certain process in the body, bone marrow, once they go and settle down in the bone marrow, that means they live lifetime. Their cells are there lifetime. So also these memory T cells, memory B cells are very important. What they do is they produce new antibodies. Whenever they see new infection, they produce new antibodies and then they get rid of the infection. And that response, the production response varies different between different peoples, age groups, health conditions, people with comorbidities. It all depends. how fast it responds. And that response comes from like long living these memory T cells, memory B cells, which is in the bone marrow also varies, right? So especially in the RNA-based vaccines, respiratory viruses, RNA-based viruses, respiratory viruses, it is kind of rare event going settle down in the bone marrow. By the time it settles down, the new variant will come out. Especially if you look at the flu viruses, every year to every 2 years, new flu virus comes out, and then we start making new vaccines as well. So the respiratory viruses, the tendency there's the fight between the virus mutations and then vaccine development, right? So that was it kind of causing to make a long-term vaccines for these respiratory viruses. But some other viruses, the vaccines are long-term effective. And there is another question. I think, how does vaccines affect the immune system? Does it suppress the immune system or to other viruses? I think this question has been already asked.
Yeah, it was answered. It was answered, sir, already.
Okay, good.
There were a few more questions are also there, sir, but since
go ahead. I mean, I am happy to answer.
Are you okay, sir? Yeah, sir. Why vaccines are not developed for HIV and HPV similar to COVID-19?
I already answered that question as well.
Okay, so then why vaccines are just working for 6 months but not for lifelong?
Just now I answered that question.
Okay, that is also fine. So then, okay, that's all. So that's all. That's all.
Yeah, I think so.
Yeah.
So one more question just showed up. Is there any side effects by taking vitamin E for a long time? It all depends on your health condition. Certainly, yes. If it is too long, I mean, there's going to be certain side effects for, like, vitamin D, vitamin E. But I would say, contact your physician, your doctor, and check up, like, do a test. How much vitamin E your body is producing right now? Is it sufficient? Do you have sufficient, or do you have too much? Do you have low? If it is low, you take it. If it is you have sufficient, I don't think you need to take any supplements for that.
So,
just get tested.
Sir, Hare Krishna, sir, would like to ask a question.
Go ahead.
Please,
go ahead.
Yeah, Hare Krishna, sir, please ask.
You're breaking up. You're breaking up. We can't hear you well.
Yeah.
I can't hear you well. You can type it in the chat window. Your voice is breaking.
Veera Somaya sir want to respond. Unmute, sir. Veera Somaya sir. Please unmute.
Sir, unmute yourself, sir. Veera Somaya sir. You're muted.
Am I audible now?
Yeah. Go ahead.
Yeah. Good evening, Mahender.
Good evening, sir. Good evening. Thanks for patiently waiting the late. It's already like, for you, it is almost 12 o'clock.
11:40 in Dallas.
Yes.
Yeah. So,
midnight.
So, congratulations, sir. We have a very nice presentation, and you have given a lot of information. Of course, it's not new for me. I have attended 3-4 lectures as well as in TV debates. We are seeing regularly in TV9.
Okay.
So Mahender ji is well-known chemist, biologist, and pharmacist. So he has gathered a lot of information, and his contribution to R&D for this COVID vaccine is appreciable. So in future also, we hope he will deliver a lecture to the students of Department of Chemistry whenever he visits the India, and we will invite you, sir, to interact with our students and faculty. And
yes, sir. I'm planning this year, sir. Certainly this year.
Definitely. We have an interaction session with the students. So because you have to motivate the students nowadays, what kind of students are getting seats in MSc, whereas in 80s and 90s used to get bright students, but because of this IT industry and everybody is joining engineering and medical side, so people like you if you motivate our students, definitely once again, the chemistry will flourish. So thank you, sir, for your eminent lecture.
Certainly.
And once again, I thank my student, Dr. Yadaya and Shukla and others for giving me chance to listen Dr. Mahender B lecture. Thank you very much, sir.
Thank you, sir.
Thank you, sir. It's my pleasure. My pleasure, sir.
Thank you, sir.
Certainly,
Thank you very
will
much,
do
sir.
what you. Certainly. Thank you.
Now it is the time to conclude. We had covered almost more than 2 hours, sir. One Dr. Satyam had raised his hand.
Hello.
Will you respond, sir? Satyam, sir?
I think Hari Krishna wants to ask a question. Go ahead and Hari Krishna. Please.
Yes, sir. Sir, we get COVID-19 in December, no, sir. December 2019. Almost we get
2 years.
We get a
2 years.
vaccine for first dose after two years, 2.5 years. So you said, as you said, the virus mutated to Delta, Beta. So is it necessary to take the vaccine of COVID-19?
Yeah, I heard only partially from you. Again, it broke down. You said it started in 2019. So I really didn't get the question.
Yes.
Can you type the question in the chat window? It's easy. And there's a couple of more very important questions
okay,
are asked. Okay. There are 2 important questions are asked in the chat window. Let me answer that.
Okay.
So the first one from Dr. S. Jansi Rani. The people who are having bronchitis problem, what will be the effect of COVID-19 on them in long run? It's a great question. It's a very important question. See, the lungs, right? Bronchitis is impacted by the lungs. Obviously, and COVID-19 targets the lungs too, right? So I would recommend them to avoid the infection. If they get the infection, they should go through, medical treatment, certainly immediately consult the doctor. And then, he has to observe what is the condition, what is the lungs condition, what is the bronchitis condition, and this infection condition. Based on, they can make a decision rather than, we can generally speaking. So it all depends on the patient's that particular condition and situation, right? So, but certainly they have to be very careful. In the long run, obviously, see, when you see the symptoms, just go and check it out. That's what I would say. But we do not know much studies about this is a 2-year-old virus right now. We can't speak about the long run, how long it's going to happen. Again, you hear about long COVID, right? Some people who get the infection, they're continuing for years, 1 year, 6 months, 8 months, 1 year. Some people lost taste. The the smell is not recovered for months, 6 months, 8 months. So we're still trying to understand what's going on with the virus impacting not just the lungs. We recently coming to know from multiple studies that it's impacting heart, it's impacting the kidneys, liver, brain. It all very mild number of patients, but it's also impacting all of these body parts, not only the lungs. So that means if someone already has a comorbidity, I would say that you have to be very cautious and careful. That's my answer. There's a second, another question from Ravi Ragam, I think. So it's another valid question. I took Pfizer booster dose in USA. When I return to India this month, can I take Covishield booster dose? Because I took 2 doses of Covishield. No, not necessarily. So if I understand your question correct, seems like you took the 2 doses of primary vaccination Covishield doses, first and second dose, and then you got the 3rd dose as a Pfizer booster. That's what I understood. If you got the 3rd booster dose, the Pfizer vaccine is the 3rd booster dose, you don't have to take any Covishield or any vaccines. You're fine, you're protected, safe. Lankela Sashi something asked me a question. One Ayurvedic medicine came and cured COVID. Now it is very crude and demanding medicine without any side effects. My question is it scientifically proved? Is any research work done on it? So I don't know what kind of Ayurvedic medicine we're talking about. I have
Anandaya medicine, sir. Anandaya medicine.
I don't call that as a medicine. And I particularly didn't go through any clinical trials. It is not a medicine. It's not properly formulated. It's not. if that is the medicine they're calling. I don't know whether it is even cured or not. I don't know any studies. Nothing is published about it. Nothing is reported scientifically. Data is collected. Data is documented. I doubt it. So when something that is not the data is not documented or published, I don't know what to call about that. I don't want to comment on that too much because it doesn't come under the treatment options. It doesn't even come into the Ayurveda. Ayurveda is a science. Ayurvedic it's a medical science, a wing of medical science. I don't think that comes under even Ayurvedic. It's just mixing some bunch of things, cooking something and makes out of I don't know. Yes, of course, chemistry is cooking. I know, but you do it properly, and you document everything, you measure everything, and you confirm what you made, what you make, what you're going to make. We have methods, right? I always say that there is a saying, the best organic chemistry is going to be the best cook always. So it's just one of the sayings. But the other cooking, I don't know what I can say.
Thank you, sir. Thank you so much for your answering the questions also patiently. Now I request our head of the department, Dr. Vipaga Shukla, to conclude the session by his vote of thanks. Meanwhile, I request all the participants to submit the feedback form. Kindly submit the feedback form. Thank you so much, sir. Now I welcome Dr. Vipaga Shukla, sir.
Okay. Good afternoon, everybody. Mahender Dewalji, I would like to thank from my bottom of heart. It is very enlightening and it was very, very effective. And you have thrown light on the evolution part and the vaccinations, everything. And I have gone through with your previous videos from basic channel, but there was a difference. I could find that you have thrown special light on chemistry, sir. I enjoyed that part. And we request you to permit us to circulate this video so that more students can get benefited out of this. Sir.
Absolutely, Shukla.
And
absolutely.
Sir,
Please
thank you, sir.
circulate
Thanks a lot,
it as
sir.
much as possible because this is
Thank
the
you,
time.
Sir. Thank you very much, sir.
Message is
Sir.
needed to people to reach the real and genuine information need to be transmitted to as many people as possible. It's most important.
Sir, thank you very much, sir. And kindly make us a promise there when you come to India, try and kindly visit our department also.
Absolutely.
Sir. Come to our college, sir. We request you, sir.
Absolutely.
So thank you very much, sir.
I'll certainly visit my department, my chemistry OE campus department. That's my first priority.
Sir.
And then my next is going to be City College.
Thank you. Thanks a lot, sir.
Thank you.
And
Thank you very much.
Thanks a lot for clearing all the doubt with lot of patience, sir. Because at the end, most of the people they lose their patience. But you have cleared each and everybody's doubt. Thanks a lot, sir. And every good thing has to come to an end. And your name is specially, sir. This is a personal note from my side, sir. Dewal indicates a temple, sir. You are the king, Mahender. Okay, you are in scientific. You are the world. You are the king, and you are doing great job, sir. Personally, I appreciate you for your efforts.
Thank you so much for that.
Concerned about the society, and on the other hand, it is bringing laurels to our chemistry department also, sir. On the other hand, I would like to thank Professor Ashok, sir, Professor Veerasomaya, sir, Professor Satyam Garu, and Budu Ramesh, sir, especially because he has given this Zoom channel for the purpose, sir. I would like to thank Budu Ramesh, sir, and TV Narasimha Rao, sir, my well friend, and Vital Rao, sir, Dr. Vijay Lakshmi, Dr. Rajita, Dr. V. Vijay Lakshmi, Dr. Sridhar Garu, and so many friends are there, sir, and eminents are there on the other hand also. And I would like to thank from behalf of Department of Chemistry and on behalf of our City College, very, very thank to everybody. Especially, I would like to thank our principal for permitting us and giving all facilities for conducting these programs, sir. And I would like to thank one and all. And last point, most important point, 3 points which we are, as a chemist or non-chemist, we should take with us are: third booster dose is very, very important to prevent death. First dose, second dose, third dose, you can prevent death, but you can face the virus. Second, boost your immunity. Naturally, try to boost your immunity. That is the second strongest point we would like to take. And third most important point, and non-pharmaceutical very, very important, like mask and social distancing, sanitizing, everything. And we have prepared a video and we will like to circulate that also, sir. And I would like to thank one and all, those are directly or indirectly helped to make this program successful, sir. Once again, I thank Mahender Dewalji for your kind gesture, for giving an opportunity to make available this, sir. Thank you very much, sir. Thanks a lot to everybody.
Thank you very much, Dr. Shukla Garu, for such a kind and nice words about me. I really appreciate that. And thank all the Department of Chemistry of City College of Hyderabad for inviting me and giving me this. Yeah, and one last question. I think this Hari Krishna, he typed the question. I would like to address that. Sure. Somebody, let me answer this question, and then whoever wants to talk. I mean, I'm happy to stay here. Don't worry. If you have more questions, and feel free. I'm here. I mean, this is a time we need, like people need it. So I'll be happy. I don't have any problem. So tomorrow is a Sunday. I don't have to wake up early. Yeah, I can stay as long as you want. So is it important to take Hari Krishna's question? Is it important to take COVID-19 vaccine now? Because virus gets easily mutated. So it seems like Hari Krishna didn't attend my talk. Probably he may have joined like in the end or something. So that's what I spoke about, right? You know, how vaccine is protecting, how the vaccine is made, what is it made of, when you have to take, where you how many doses you have to take. That's what all I talk about. So when you get a chance, go and watch this in the YouTube. You will get an answer to your questions from my talk. Yeah, I think there's
Sir, I think it is done, and I would like to thank each and everybody, and the session is concluded, sir. Thank you very much, sir.
Thank you.
Thanks everybody.